Hi everyone, my name is Diogo and I'm a postdoc at Professor Gordana Vujanović's lab at Columbia University, and I would like to start by thanking the organizers for this opportunity to present the work that we've been doing in our lab on organs on a chip and drug development. We are extremely interested in drug development, which is a very long, expensive, and highly inefficient process. And when we look at anti-cancer drugs, it's a worst-case scenario where only 1 in 15 new cancer drugs that go through clinical trials receive a full approval from the FDA and enter the market. In our team, we believe that this is largely due to the lack of predictive preclinical testing models. The animal models, cell lines, and other preclinical models that we've been using for years, they're very useful and they give us a lot of insight, but they do not fully recapitulate human physiology and pathology. So our team believes that organs on a chip, the microphysiological platforms that we are here meeting to discuss, may lead to a faster and more affordable development of new drugs. A couple of years ago, our team presented a dual tissue organ on a chip with human cardiac tissue, home-bone tumor tissue, and then we recapitulated the safety and toxicity profile of lincitinib, an anti-cancer drug. When this drug was evaluated in preclinical models, it was safe, it was not safe, but it was highly effective. And then we jumped to the clinical trials. We observed the other side of the coin, where it was ineffective, and it didn't have as much cardiotoxicity as in the preclinical models. When we did the same regimen in our chip, we observed the same results as the clinical trial. So we were able to recapitulate in vitro using our platform the profile of lincitinib. So based on this experiment, we took on ourselves to increase the complexity of our model so we could ask more complex and different questions. So we went from a 2-tissue chip to a 4-tissue chip, where each tissue can live in their own individual niche. We'll have in the bottom a selective permeable endothelial barrier that will separate the individual tissue niche to the vascular channel on the bottom. And then we have a reservoir at the end where the perfusate can mix and where we can add new drugs to the system. Our chip has the size of a glass microscope slide, and it has an open setting, allowing for the collection of media and also microscopic visualization throughout the entire experiment. We've been culturing 4 tissues in our chip: liver, heart, bone, skin, and an endothelial barrier at the bottom of each tissue chamber. These tissues are generated from human-induced pluripotent stem cells, and they are formed and matured in their own custom-designed bioreactors. By doing this, we are able to generate adult-like phenotype tissues, where they will recapitulate some of the known adult-like phenotype functions that we see in people. And by also culturing them in their own custom-designed bioreactors, we can troubleshoot, validate, and quality control the tissues before we transfer them to our multi-organ chip for integrated studies. Our plug-and-play chip has a modular design with individual tissue chambers and has open access, as I mentioned earlier, for then real-time imaging and super data collection, and has the perfusion channel at the bottom, which is lined by a selective permeable endothelial barrier. Yesterday, Casey Ronaldson-Bouchard presented this chip with more detail and presented several of the applications that we've been using, and today I'm going to discuss how we can use this chip to recapitulate doxorubicin PK and PD. Doxorubicin is an anti-cancer drug which is used to treat several different types of cancer. However, it has a long list of side effects which prevents its wider use. So we added a clinical relevant concentration of doxorubicin to our reservoir, and we tried to see if we could recapitulate its PK and PD in our chip. We compared 3 different experimental groups. On top, the multi-organ, where we had 4 individual tissues: liver, heart, bone, skin, cultured in an integrated setting with an endothelial barrier at the bottom. The mixed group, where we had the same tissues: liver, heart, bone, and skin, but without an endothelial barrier, so with time the individual media will just mix into a common type of media. And then in the bottom, the isolated, where we culture each tissue individually without communication with any other tissue type. Then we look into the pharmacokinetics, how the drug is absorbed, distributed, metabolized, and excreted in our system, and the pharmacodynamics, how the tissues are affected by this drug. Starting with the pharmacokinetics, in order to better model Doxorubicin PK in vitro with the help of CFT research, we developed a mechanistic multi-compartment model for in silico simulations. We modeled the reservoir, the entire tubing system with a pump, the vascular channel at the bottom of our chip, and then the 4 individual endothelial barriers at the bottom of each tissue chamber. We also model sampling out of each chamber. We model also the exterior of the tubing to take into consideration an absorption that could happen from this tubing. And we model each tissue chamber individually, where we took into consideration the geometry and the topology of each tissue chamber, of each tissue. Starting with the endothelial barrier, we model it by generating a three-layer tissue where we have a porous membrane in the middle, and then on top an MSC layer, and in the bottom a UVEC MSC. In the liver tissue chamber, we model the liver tissue in the bottom of the tissue tank where it normally rests, and we model the tissue tank in 3 different layers. We observed that when we modeled our tissue tank into these 3 different layers, we could better replicate the drug distribution in the system instead of having a 1-layer, well-stirred tank. We did the same for the heart, where we modeled the PDMS pillars that it's hold on, the bone, and then the skin, keeping in mind that the skin needs an air-liquid interface to have an adult-like phenotype, so we model also the skin on top of our tank. We finished by modeling the entire reservoir and tubing system, and in order to reduce the computational necessities of it, we divided it into 20 individual subcompartments. So what happened next? We introduced the drug into our system, and at 1, 24, and 72 hours, we collected samples from each individual compartment. We ran spectrometry analysis, and then we looked into the constitution of doxorubicin and doxorubicinol over time. We compared experimental data with the PK model, and we observed there was a very good correlation between the 2 different models, where we saw an increase of the concentration of doxorubicin and then a slight decrease over time in all tissue chambers. Regarding doxorubicinol, which is the main metabolite of doxorubicin, and it's generated in the liver, we observed its increase over time in all tissue chambers. So it was being metabolized properly, and then it was being distributed into the chip. This is in the multi-organ chip, where we had the endothelial barrier at the bottom. When we looked at the mixed chip, so no endothelial barrier, we observed that doxorubicin has a much higher, much faster peak at the beginning of the experiment, but then still decrease over time, and we observe a very good correlation between PK results and experimental data. Same thing with doxorubicin, higher and faster peaks than into the into the InterOrgan group. When we compare both, we can see this better, that the mix in red has a much higher and much faster peak than the InterOrgan, and same thing with doxorubicinol. We believe that this happens because without the presence of endothelial barrier, there are less barriers for the drug diffusion in the system. So after looking at PK and seeing that we could correlate the experimental data with in silico data, we looked into PD. Could we replicate doxorubicin's PD in our system? And starting with the liver, we looked into tissue-specific readouts. Starting with albumin on top, we see that exposure to doxorubicin led to a decrease in albumin production, but a stable production of urea, which correlates with clinical data in patients that have doxorubicin as their treatment. When we moved to the heart, we saw there was an increase of the cardiac troponin I in the groups treated with doxorubicin, and a decrease in beat frequency in the same groups, which also correlates with patient data. We then moved to the bone, and we observed there was a decrease in bone cielo protein release in the multi-organ chip, a decrease of cellularity in the same chip, and stable trap activity, which makes us think that osteoblasts are more affected by doxorubicin osteoclasts, which also correlates with clinical data. We finalized by looking at the endothelial barrier, and in the InterOrgan chip, we observe a decrease of resistance in every endothelial below the liver chamber, heart, bone, and skin. So we were fairly able to recapitulate doxorubicin PD in our chip. So we asked ourselves, can we use this system to identify biomarkers of doxorubicin cardiotoxicity? Because what we observe is that a lot of patients that have doxorubicin, then they have cardiotoxicity and heart failure years later. One of the current biomarkers is cardiac troponin I. They observe that patients that have higher levels of cardiac troponin I after doxorubicin treatment will have heart failure, or most likely to have heart failure in the future, and we observe the same results in our chip. We then look to mRNA, and mRNA-1 has been recently identified as a potential biomarker of doxorubicin-induced cardiotoxicity, and we observed that it was upregulated more in the multi-organ chips than in the mix in the heart chambers, but also in the perfusate. It was upregulated just in the perfusate of our system. Next, taking into inspiration a clinical study that identifies 17 biomarkers of cardiomyopathy. Following PCA, we observed that the mRNA from our multi-organ 4-tissue chip cluster closer to the clinical data, unlikely the mixed 4-tissue platforms, making us believe that the presence of endothelial barrier in our chip and the separation between tissues and the vascular channel is important to fully recapitulate the cardiomyopathy observed in patients. We then look at the mRNAs that were more likely to be associated with cardiomyopathy, and we saw that the fold changes in the multi-organ 4-tissue correlated 85% with clinical data, while the mixed, where there was no endothelial barrier, only correlated in around 29% of the cases. In conclusion, in this study, we generated an in silico multi-compartment model of our chip for studies of doxorubicin PK, and we observed that this computational model closely matched the drug's PK in vitro. We also demonstrated significant correlations between doxorubicin's PD profile in the chip and clinical data, and finally, we were very excited to see that we were able to identify biomarkers of cardiotoxicity and cardiomyopathy. What is next? What we're going to do after this? First, we are going to add other tissues that can influence the drug PK and PD, because right now we only have liver in our tissue chip. We believe that by adding gut, fat, kidney, we'll be able to better recapitulate the drug PK, PD in our chip than the ones we just deliver. We also plan to test different types of drugs and routes of administration and different dosing schemes. For example, with the presence of the skin tissue in our chip, we can test topical drugs and see how they are absorbed, distributed, and metabolized on the chip, and which effects they will have in the other organs. And finally, we also are planning to develop a more scalable version of the chip to allow automation and compatibility with high-content imaging systems and instrumentation. So this chip can be used in other academic centers and industry partners in a replicable way. I would like to finish by thanking the entire team at Gordana's lab, our collaborators at Columbia University, the CFD research team, which was instrumental to help us develop our computational model, and then our partners at UV and BU. We also like to thank our institutions and funding support. Thank you very much.